Okay, so in the last class we have seen the different terms which are used in the chapter genetics, that is inheritance and also variations, and they are very important to understand the different concepts which you are going to see. And also we have also seen the different characters which have been studied by John Gregor Mendel in using or in the case of pea plants. With that idea, let us see, and I think I made a mention about some of the results which were obtained by Mendel during the experiment. And what was the experiment conducted by him? What is the experiment? Yes, anybody? I think you can see this one. So we have seen that when we take one gene, if you take, so we have seen can you hear me?
Yes, sir.
And depending on, in any plant or any organism, thousands of characters are there. But is it possible for us to study all the 1,000 characters at a time? Is it possible? Huh? So definitely it is not possible to study all the 1,000 characters at a time. So that's why what is being done here. So we are going to study either one or two or maybe only few characteristics are going to be studied here. So depending on that, suppose if you are studying only one trait at a time, it doesn't mean that other traits won't be there. But only thing is that we are studying only that trait, that's all. Other traits will also be there. So such cross is what we call it as monohybrid cross. What is this cross called? What is this called? Called monohybrid cross. You can see this, So what is this monohybrid cross here? The cross where one character is studied here. One character, or you can say trait is studied. Then if you are studying two traits, what is that called? Two characters or traits are studied here. That we call it as dihybrid cross. So monohybrid cross and also dihybrid cross. These are the other technical terms which we are using here. Suppose if you are having three, that is called trihybrid cross. So where three characters or traits are going to be studied here. Like this, we are going to use different things here. In the last class, I think I made a mention about one of the trait here. You can take any character among those 7 pair of contrasting traits which was studied by Mendel. So I think we took the example of, if you take the parent here, so parent, in this we have seen the male parent. So what is the symbol here? Can you see this one?
Male.
You can see the symbol, one which I have written on the board.
Yes, sir.
This is female. Okay, male and also female we are going to use here. And in this we are having the tall plant and also, let me say, dwarf plant. Tall plant and also dwarf plant. Then the question is, so when this crossing was done, crossing means, what do you mean by crossing here? So selectively it is to be bred here. Selectively it has to be bred here. So different techniques which we follow during the hybridization. What are the different techniques which we follow during hybridization? Second chapter Lodit Bola. Which are the techniques which you follow during artificial hybridization?
Bagging.
Huh?
Emasculation.
Good, very good. Emasculation, bagging and tagging. So where unwanted pollen rings are going to be removed, unwanted anthers are going to be removed, and then the stigma, or you can say the pistil, is going to be protected from unwanted pollination here. So that is what the selective breeding or artificial breeding, that is what is done here. So we want to have a particular plant to get pollinated, the soil. So for that we call it as artificial hybridization. That was done between them here. So two different plants. Okay, so then for in that they took one plant with the tall, that is the height character we have taken, and also another plant which is dwarf in nature. Then what is that which we expected here? We expected it to be a mixed character here. Mixed in the sense, mixed again So mixed blending, combine. Blending it is also called blending. Mixed characters we expected here, but there was no blending of the traits here, no mixing up of the traits as you rightly told. But normally we use the technical term called, what is the term called? Blending. Okay, so no blending, and we got, so he got only tall plants in the F1 generation. It is said to be F1 generation. He got only F1 generation. So what we expected, suppose if you are doing the same experiment, you think of yourself as a scientist. You have started an experiment like this without knowing the results. Since you are knowing the results, we have been speaking about it, you are telling now. So, if you just think, if we are the scientists, definitely when we do that, what is expected from the crossing between the tall and also dwarf? What is that which we expect? Can expect what?
Medium height, sir.
So we expect medium height here. So that is what is called blending. But for the surprise of Mendel also, he was also surprised. So there was no blending. There was no blending of traits. So there was no blending. That's the thing. Then he went for the selfing here. The next process is selfing. Selfing was done. In this selfing, parent F1 we have got. So selfing between F1 and also F1. F1 into F1. What do you mean by this? What do you mean by this? So he took some two plants from the plants which you obtained in the first filial generation. First filial generation. We made a mention about filial generation also. So in F1, that is first filial generation, he got tall plants, and the same tall plants were taken here. F1 into F1 means, so that is going to be, so tall crossed with and the top plant. So what is that which we expect here? So this give rise to, so he got some results in the next generation, that is F2. We call them as F2 hybrids. F1 hybrid and also F2 hybrid here. So both parents are tall. What is that which we expect them?
Tall, sir.
Tall. Both should be tall here. But to his surprise, so he was able to see the plants, so which were some were tall plants and also dwarf plants, and that too in the ratio. So tall plants were there and dwarf plants were there in this. So both were obtained here, and that too in the ratio 3:1. That means 75% of the plants were tall and 25% were dwarf in nature. So then how is it possible in this case here? How these things are possible. For that, he gave an idea. So he tried to explain this cross with the help of the genes. You know, in a gene, yes? Yes, anybody?
Due to dominant.
Yeah, that's what. So that is 1 minute. He tried to explain that one. Let us see how he was able to explain the things. In this, what he did here. So, this he tried to explain with the help of the. He spoke about genes. Already we know what are these genes. What are these genes? What are the genes? Hey, genes on that. Anybody?
Functional unit of heredity, responsible
Functional unit of
expression of characters.
huh?
Responsible for expression of characters.
The character
Actually, these genes, according to Mendel, these genes are nothing but factors. He used the term factors. So genes are nothing but the factors. According to him, what is the explanation by Mendel? So where you can see 3:1 ratio is there. That means 3/4 of the plants were tall plants and 1/4 of the plants were dwarf plants. So mukka vaga tall plants or kal vaga dwarf plants banta. And he used the term factors. So of course he didn't use the word genes, later on only we have been using this genes. So that was given by other scientists. And he used the term factors. So now we call them as genes here. Genes are nothing but the factors. So according to him, these factors are the stable ones. These are the stable factors. They are very stable factors. And so these are the things which are responsible for traits. Responsible for traits or characters. And in these genes or factors you have. So he told that there are some contrasting genes, contrasting factors, or what is another name for factors? What is another name for factors?
Genes.
Very good, genes. And these contrasting factors or genes are said to be alleles. Alleles are allelomorphs. They are called alleles or allelomorphs. Alleles are allelomorphs. So he tried to explain by giving the names in this case here. In this, he used the, for example, if you take, so whenever you are writing the cross here, so this, what do you call this cross where only one gene is involved? What do you call the cross where only one gene is involved? Monohybrid cross. So, according to him, as he rightly told, in these alleles we have two forms here. These alleles are having two forms. So one is said to be the gene or allele which is said to be dominant allele. Dominant allele. The allele. So there are two different forms which are responsible for, which are contrasting. Contrasting means, what do you mean by contrasting? Very good, opposite. Okay, so one allele or one allelomorph is said to be the dominant allele and another allele is said to be recessive allele. Recessive allele. So these things you should understand properly. Okay, if you have any doubts you should speak. Because these are all that lot of technicalities are involved in this. If you have any doubt, you please ask. So, yes. ಏನಪ್ಪ? ಅಲೀಲ್ ಅಂದ್ರೆ ಏನು ಅಂತ Okay, alleles or allelomorphs are nothing but two different forms of the same gene. So you are going to market. Okay, so when you go to market, let me say you are having a banana. So there in banana, one may be highly ripened, or one may be very small, one may be very big in size. Or when you go to purchase some apple, you may be having very big apple and also small apple. So both are called apples, you know. Do you call them as apple or not?
Yes, sir.
Definitely yes. But the only difference is that, and most of the characters, 99.99% you can say, of the characters remain same with little difference here. So with reference to size of the apple, if you say the bigger one is there and smaller one is there. These are the two forms of same apple. Do you agree with me? Do you agree with me or not? ಆಪಲ್ ಅಲ್ಲೇ ಒಂದು ದಪ್ಪ ಸೈಜ್ ಇರೋದಿದೆ, ಇನ್ನೊಂದು ಸ್ಮಾಲ್ ಸೈಜ್ ಇರೋದಿದೆ. ಅಂದ್ರೆ ಎರಡು ಆಪಲ್ ತಾನೆ.
Yes, sir.
In the gene also, the gene is the one responsible for a trait, for a character. In that we have, just like in the apple we have seen the small size and also big size, which are opposite to in nature. ಎರಡು ಕಾಂಟ್ರಾಸ್ಟಿಂಗ್ ತಾನೆ ಆಪಲ್ ಅಲ್ಲಿ ಸೈಜ್. ದೊಡ್ಡ ಸೈಜ್, ಸ್ಮಾಲ್ ಸೈಜ್, big size and small size. Like that, here also contrasting it is opposite. So, but the gene is responsible for a trait. In that some differences will be there, very minute differences will be there, and so that's why it is alleles are said to be two different forms of the same gene. I think I told you last class also, if I am correct. ಹೇಳಿದ್ನಪ್ಪ, ಹೇಳಿದ್ದೀನ್ರ ಬರ್ಕೊಳಕ್ ಹೇಳಿದ್ನ.
Yes, sir.
So alleles are the two different forms of the same gene. Alleles are the two different forms of the same gene. Barkodedana. That's very important. This is a good question. If you are having doubts like that, you should ask. Okay, you got it here? Artha aitanappa? Any doubt there? So we have the two different forms here, and these two different forms are said to be dominant allele and also recessive allele. Dominant allele and also recessive allele. And always this dominant allele, for example, if you take the height, if you are having height here, if you are having, so there are two different alleles here. The dominant allele is said to be capital T, and the recessive allele we call it as small t. You can use any alphabet, but only thing is that I will be sharing the last class this one. Okay, so you in that I have given you more so technical terms. Definitely you will be benefited. If you have any problems, you can say speak to me also later on. Okay, so somebody, I think they are finding it a bit difficult because of the technicalities which are involved. Definitely, especially these two chapters, you know, if you are missing the classes, it makes you difficult. Okay, because the flow is there, continuity is there, and one is linked to another one here. Once you understand the things, it is very easy to present the things also. Okay, so they are all going in a flow. It is in a flow, it's going. Okay, one link is connecting in it. If you find any difficulty, I can help you. Don't worry. Okay. So then coming to the issue. So capital T and also small t we have given here. Like that, you can use, suppose if you want H also you can use it. Capital H, small h. Only thing is that always the dominant allele is going to be shown as capital T and the recessive allele is going to be shown as small t or small letter. Capital letter is used to indicate the dominant allele and recessive allele is indicated by using small letter. That is the one rule which you should always remember here. So in this here the dominant allele in capital letter and recessive allele in small letter. And for a character, for a character, how many genes, how many alleles should be there then? How many alleles are controlling one character in an individual? Anybody any idea?
No idea.
One character. How many alleles will be there in our body, in our cells? Two alleles. Why do we have two? Very good. Why do you have two alleles?
One dominant printer and one recessive.
Two alleles. Should it be same? Same. So here we are having tall plants here. Tallness. So should it be only the dominant alleles which are there here? So in any individual, if you take any individual, you know every cell is having the same DNA in our body. Do you agree with me or not? Do you know it or not? So same DNA is present in trillions of cells which we are having. And out of that what happens here? So how many set of chromosomes are present? How many set of chromosomes are present in an individual? Like us? Huh? How many sets? 23 sets. 23 pair means how many sets? How many sets of chromosomes? What about the ploidy? Ploidy is nothing but the set.
2n.
2n. Why? Why do we have 2n? That means two set of chromosomes here. In the sense, we have the chromosome. Last class I think I have shown you. We have a chromosome like this, one chromosome, and the same way we have another chromosome. Last year ಓದಿದ್ದೀರಾ cell division ಅಲ್ಲೆಲ್ಲ. See, do they look alike? How many chromosomes are here? How many chromosomes are here? ಎಷ್ಟಿದಾವೆ? 4 ಇದಾವೆ. ಕಣ್ಣು ಬಿಟ್ಟು ನೋಡ್ರಿ ಅಲ್ಲಿ ಸ್ವಲ್ಪ. 2 chromosomes are present. This is one chromosome. This is one chromosome. This is another one here. Okay. And in this, so do they look alike? How do they look here? They look alike. They look same. They look same in this. And such chromosomes, what do you call such chromosomes which are looking alike or same? ಏನಂತೀರಕ್ಕೆ?
Homologous.
Very good. They are called homologous chromosomes. What are they called? What are they called? Homologous chromosomes. So here, you know that there are two alleles which are going to be present here. If you are getting confused, you can show only one chromatid also. That's not an issue. Only one chromatid also you can show here. And we have a place where the genes are located. What do you call them? ಏನಂತ ಲೊಕೇಶನ್ ಅನ್ನ, ಜೀನಿನ ಲೊಕೇಶನ್ ಅನ್ನ. What do you call that one? That is called, what do you call the location where the genes are present or alleles are present? Third one. What do you call this one? That is called locus. That is called locus. So we have, are you getting me or not? Are you getting it or not? ಅರ್ಥ ಆಗ್ತಿದೆಯೋ? If you are getting confused, you can rub it off. This happens because of the duplication of the chromosomes. Duplication ಆಗುತ್ತೆ cell division ಗಿಂತ ಮುಂಚೆ, ಹಾಗಾಗಿ ಡಬಲ್ ಇರ್ತದೆ. And you know that only during cell division they are going to be present in the form of chromosomes. Otherwise they are going to be present in the form of chromatin network. Just for your easy understanding, I am showing you only one chromatid in each chromosome. Okay, don't get confused. Ingar artha agta ide iga. And here this is said to be the locus. Let me say there are two alleles, allelomorphs, and two forms of the same gene are present here. It can be capital T and capital T. Okay. It can be capital T and capital T. And so this the second gene chromosome is there, that is the exact copy of the first one. That's why we call it as homologous chromosome. What is this called? Homologous chromosome. So that is a homologous pair of chromosomes here. That means how many alleles are going to control the trait in an individual? ಎಷ್ಟು alleles ಇರ್ತದೆ? 2 alleles are going to be present in each individual and one each on one chromosome, because we have 2 chromosomes with the same structure, that we call it as homologous chromosome, and that's why 2 alleles will be there. The 2 alleles can be saved, as we have seen in, for example, if we are taking this, both dominant alleles can be there or they can be recessive alleles, both can be recessive alleles, and such condition where both alleles are going to be of same type, that is dominant alleles or maybe recessive alleles, is what we call it as homozygous condition. What is that called? Homozygous. What is that called? In homozygous or that?
Same, sir.
So, if the alleles are of the same kind, if the allelomorphs are of the same kind, if both are dominant or both may be recessive in nature, so then that condition is what we call it as homozygous condition. And if 2 different alleles are there, can you tell me the combination in this? 2 different alleles combination?
Capital T, small t.
Capital T and also small t. Capital T and small t. Where you can see capital T and also small t, both different combinations here. So one is dominant allele and another one is recessive allele in this case. So such condition is called heterozygous condition. What is that called? That is called heterozygous condition. So like this we are having the homozygous condition and also heterozygous condition here. So got the meaning of homozygous and also heterozygous condition and also the alleles meaning. Alleles godaitalla meaning?
Yes,
It is a repetition of the last class. Okay, ಯಾಕೆ ಕೆಲವರು alleles ಅಂದ್ರೆ ಏನು ಅಂತ ನಾನು ಮುಂದಕ್ಕೆ ಹೋಗಬೇಕಿತ್ತು. So last time I didn't explain like this. By using PPT I had to explain the things. But I think today it makes it a bit clear when compared to yesterday, if I am correct. So we are using different terms in this case here. The gene, that is nothing but the one which is responsible for a particular character, a sequence or a segment of the DNA, that is a piece of DNA responsible for one trait or one character. And in the genes we are having 2 contrasting traits. So the factors which are responsible for the 2 contrasting traits, which we are called, which we call them as alleles. Of course, it was given by Gregor Mendel. So alleles, and these alleles, there are 2 different forms. One is said to be the dominant allele, and another one is said to be recessive allele. And suppose if both the alleles are present, which are similar in kind, then that condition we call it as homozygous condition. And if both the alleles are different, then that is called heterozygous. Suppose if both the alleles are dominant, then what do you call that one then? If both the alleles are dominant, capital T is there. What can be called?
Homozygous.
Homozygous. And what is the nature of the allele? Allele is dominant. Allele is dominant. That's why it is said to be homozygous dominant. Not in our one extension marta hogtideni. Basically again what is homozygous? Now again based on the type of allele, if both the alleles are dominant, we call it as homozygous dominant. What is that called? ಬರ್ಕೋಬೇಕಾಗೋಣ. Capital T is homozygous dominant. And what about the what about small t small t? What about small t then? You are correct. You are correct. What is that? Homozygous recessive. It is said to be homozygous recessive. So like this we are having these different forms here. So actually we have to discuss about how exactly he was able to explain this one by using all these terms. ಇವುಗಳೆಲ್ಲವನ್ನು ಬಳಸಿಕೊಂಡು ಯಾವ ರೀತಿಯಲ್ಲಿ ಮೆಂಡಲ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ರು ಅನ್ನೋದನ್ನು ನೋಡಬೇಕಲ್ಲಿ. So we have to see how exactly he was able to explain this, how one trait or one gene is responsible for one trait, and what are the rules under which the traits are going to be inherited from parent to the offspring that we have to see. Okay, so with this background, I think we will be able to discuss about how one gene is responsible for one trait, or how one gene is inherited from parent to the offspring. That we will take up in the next class only, I think. Now I think it will not be possible to complete, so we will take up that issue in the next class. Okay, any question here? Any doubts? Any doubts you are having? Huh?
No, sir.
Anybody any problem?